Thanks, Alexa. Thanks, Alexa, for the kind introduction. And I have to say, you have to add one more thing. Like, I'm also really cheap. Like, 'cause, we have to build things cheaply. So there are a lot of things that we do that can enable, more people to do things even for very low cost. Yeah, so the picture up here is our campus. So we're part of UBC, and UBC has two campuses. One in Vancouver, is the main campus, and this is like the new campus in the Okanagan. It's kind of like a winery place with mountains and lakes. And so if any of you travel in the area, please let me know, and I'll be happy to give you a tour. All right, so I think, I generally pull up a few slides to talk about why, force is important, but I don't think this is a very, necessary thing for this group. But, like just to give you the gist, like this is how I introduce things. Like, force are important because cells are not balls, right? And so give you a picture of this, tension really stretches the membrane so that there is this structure and the shape and drives the function of the cells. And at the same time, there are collective movements and emergent behaviors, and all of these are things that don't happen if you have just like random forces, random motions. But these collective motions have directions and patterns, and those are really driven by mechanical forces. And therefore, it's really important for us to understand kind of the nitty-gritty details of how the forces are transmitted from single molecule to cell all the way up to the scale of tissues. And so, you guys all know that cells experience all sorts of physical forces from the environment. And primarily, what we're interested in are kind of the forces at cellular interfaces. So this is like cell-cell interface, cell to matrix interface. And if you think about at the single cell level, there are all these mechanoreceptors to sense all sorts of different type of mechanical stimuli. But the ones that we are primarily focused on are kind of the adhesive receptors where we can potentially do perturbation and observation at the single molecule level and to kind of understand the mechanisms and potentially control those things that's happening. And the reason why we choose this is because, you can, interface is ultimately exposed to the outer environment, right? So if you add tools that can directly get into those gaps, then you don't have to modify the organism genetically if you want to study any behavior. So you can study things maybe more broadly. So this is what my lab generally does. Like we're interested in generally the mechanobiology, how to modulate adhesion, how to image molecular force. We were getting into some immune cell mechanobiology, but really we're trying to look into these days is collective and emerging behavior. And so the kind of toolbox we built is at the interface. So if you have adhesion molecules attaching to some other molecules either on the cell surface or substrate, we can replace that interaction or with a DNA toolbox with all sorts of DNA probes that you guys are familiar with. And so, but I think one of the key things that we are trying to do is to create more force responsive DNA architecture so that offers more controls or different kind of properties that can be further used towards building up this toolbox capabilities. Right. So these days we're looking into the nanomechanical architectures, like how to create those things, how to engineer energy landscape of these molecules to have special properties. We're looking at topological knots as just one of the examples and thinking about state machines and mechanical logic and ultimately how these things can be interfaced with the cells. And simultaneously, we're also doing some technique development in order to characterize a lot of these things because our predictive power is limited. So having tools to do actual experiment validation was useful. And all of this is kind of built on single molecule biophysics from force spectroscopy to single molecule fluorescence to super resolution microscopy. So that's kind of like generally the areas that we're working on. I'll talk to you guys about a few of this today. And so there are generally three topics I want to talk about. One is the quantitative force imaging with DNA, and then the second one is how to program novel DNA nanomechanics. and I'll showcase kind of a new force spectroscopy technique that we built, and hopefully this can be widely adopted by the community. So, so you guys all know that, adhesion structures are very complex, right? And so people have done a lot of super resolution stuff to look at, the 3D structures. And so these are from a structural perspective, how things are arranged. And structure always we want to relate those to function. And so, in the past few years, I think Khalid is one of the first ones who kind of built this super resolution tension sensor from a DNA hairpin. I think a lot of you guys are familiar with the concept, right? So if you have a DNA hairpin that can be opened up, then you can basically expose a previously hidden site and you can paint that. And so in addition to just the structure of adhesion, you can now image the function, right? Like the actual force. We think about force as the kind of the function of these structures, and seeing where those forces are quite important. And there are some like other people, including our group, doing different kind of super resolution imaging, both in the focal adhesion areas and also in kind of rolling adhesion. And so, when we think about force imaging in a broader sense, right? Like, what do we want to achieve? Like, we want to get very high spatial resolution because you have like local clusters of molecules and so on. So you want to resolve things at least, kind of on the order of 50 nanometers. You want to have some good temporal resolution because these structures are very dynamic, right? So you want to see how they move. If you do super resolution and they're taking an hour to kind of resolve, then you lose all that information. Ideally, you want to actually resolve the force as well, right? Because a lot of times when we think about these, DNA hairpin probes or TGT, they're kind of an on-off binary type of probe. So they report the presence of a force, and maybe there is a threshold assigned to that, so you can sort of know quantitatively, are there a force above or below a certain threshold? But can you get more resolution in the force amplitude is a question. And lastly, you want to do this all in live cell, and all of the four requirements are kind of constraints on each other because like you want to get more information, but there's only so much information, and so how do you get the most out of this while keeping the cells alive? And so I'm not sure if I need to introduce this. I think this is a concept that a lot of people are already familiar with in this community. But DNA paint is the kind of super resolution microscopy method that we use. It relies on having imager strand, which is fluorescent, uh, binding to its target and staying there for kind of a while the rest of the imagers are diffusing in the background to see these things, right? So if you have a structure like this and they're densely labeled by flow force, if you turn on one of them at a time and you localize individual ones, you can essentially paint by dot the final image at a higher resolution than the optical resolution itself. And so for those who haven't seen a DNA paint before, this is roughly how it looks like. On top is a wide field image of, like a thing, just let's call it a thing, a structure. And then at the bottom are if we record this over time. So you basically make a spatially dense object temporally spatially sparse by stretching out in temporal direction. And so by doing that, you can essentially paint something with higher resolution. And so one of the problem with DNA paint is that doing it fast is difficult. And we have been doing this primarily to expose for a long time is because all of the DNA imagers are diffusing, right? And so they're diffusing the background. But if you have a very fast shutter, you will capture all of them. So the trick that people do is to have long exposure. Right. And so it's only when you have like, the target binding to, I guess the imager binding to the target, and then they're not moving, and then you have a long exposure, then you can blur out all of the diffusing images in the background and actually see a high signal to noise ratio. And so this basically requires you to have a long exposure and relatively slow imaging speed. And for super resolution, speed is very important, especially if you try to resolve the dynamic of things. You want to accumulate as many spots as possible, as fast as possible. And so people have been trying to improve the speed by many different means. either to improve the signal to noise ratio, K on, and so on. And just around the same time we did, there's also like this fluorogenic approach. And so I just want to also say that in addition to speeding up, imaging things at interface between cell and surface also create other type of problem. And for example, if you just have a normal DNA paint imager, if you have targets on the surface, they can diffuse in 3D. So they can diffuse in and out of the turf volume very quickly, right? So you don't see them as much. But if you have a confinement, between the cell, spatial plane, and the surface, then all of your images has to diffuse within this layer into the target. And so if you're imaging fast, you actually see all of those things diffusing around. And so the ideal case is really if we have something that's fluorogenic, right? So something that only becomes fluorescent when it binds to its target. That way you don't see anything unless they're bound. And so by doing so, you can actually achieve higher imaging speed because the exposure time no longer becomes a problem. And so we took a very simple approach. And so we took a molecular beacon. We're like, oh, why can't we just use that? And we tweaked it to have very short stem and very long loop, so that the two ends are very close and they're quenched when they're not bound to the target, and they're separated when they are, right? Uh, and so just very briefly that, you can see that number one, they, at equal molar as normal imagers, they create almost no fluorescence background at all, even at 200 nanomoles concentration. And you can actually do, epifluorescence imaging mode with this because, basically the background fluorescence is almost nothing. And so we did some comparisons for spatial resolution and showed that they're very similar to linear imagers. And how do you image with tension is exactly what Khalid's lab has pioneered in the past. It's a DNA hairpin probe. Actually, I think we use the exact sequence. So, a DNA hairpin probe would open and close around the F half value, right? And so that opens up, and the red part would be exposed, and then that would bind to this to show a spot, right? And so the concept is very similar. Force creates open state, and the imager will bind to the open state. And I would say like one of the difference is that we're actually binding to a part of this that has tension, and we'll show you in a bit why that's actually somewhat useful to have. And so here's just a movie of, like a DNA paint of three cells spreading over time. This is all sped up, but you can sort of see, how they work. This is with the molecular beacon tension probe um, imager. And so you can sort of reconstruct how these things look like, where the tension events are. And what we show here up here is if you're doing this with just linear imager, you basically, if you image fast enough, you basically see all these things diffusing under the cell, and if you do it at a particular speed, they all contribute to kind of artificial localization event under the cell. But if you have the molecular beacon imager under the cell, you basically see nothing at all. So basically, this helps you remove the artifacts from confined diffusing imager during fast imaging. And so the next question we have after this is like, can we quantify the force in tension imaging, right? So with this, the force is essentially we're reporting is force above or below a certain threshold. But can we get an amplitude out of that? And so the idea is that, previously when we tried to image things, with DNA paint, we're trying to image close to the kinetics of the unbinding event, such that you get the most photon yield per frame, and there's not too much point to image much faster than that. But the problem, of course, is that, if you can image faster, then you can get much better information about the unbinding kinetics, the lifetime of the bound state, right? And so people have been doing similar things to get kinetic parameters from quantitative paint, and we're like, can we use this here? Can we encode or decode force from the binding kinetics? And the answer is yes. And there has been a prior study using optical tweezers to look at the unbinding kinetics of a short oligo by exerting attention on this red single-stranded DNA and see how long this blue one bind and unbind. Right? And what they found is that as force increase, the K-off becomes, exponentially faster. And so I think traditionally we didn't want this partly because when you have a high tension, you don't have enough kind of event that's visible, so it's hard to visualize. But because we're now using the molecular beacon, I think the signal to noise ratio got a bit better, and so we can actually start to detect those K-off event even at very high forces and higher speed. And so the idea is that if we can figure out what the K-off is from those DNA paint movie, then we can map it back to the force that is being exerted. And from this study, we modeled it and we showed that the K-on was not as much influenced as K-off. The K-off is kind of 4 orders of magnitude going from 10 to 40 piconewton, but from 10 to 40 piconewton, K-on was only changed by a factor of 2 or 3. So it's not as bad. So you still have the painting events, maybe a bit less, but you can get some more information. So the detection scheme is exactly the same as before, but simply we're trying to do a bit more information extraction from this type of experiment. And so here's a movie of one cell that is like over time, like a raw paint movie. You can basically do all the localization of events, and then you can now do single molecule tracking to link these events together. And then once you do that, you can then look at individual tracks of one molecule and build a kind of a series of like kinetics of that one molecule. And so here it's a kind of a 3D representation over time of all the binding events. So there's a lot of information we can now extract by looking at the I guess the temporal dynamics. Here are 4 traces of really fast events versus something that's really slow. And then with enough statistics, you can basically get kind of a force value with kind of the error bars on those. And so we can probe kind of a range of forces with this example, something like 20 to 30 plus piconewtons. And so with this kind of construction, you can go from the normal white field to tension paint, but then with the additional, I guess, kinetic information, now you can start to map out what are the average force acting at that spot over a range that's roughly from 9 to about 30 piconewton. And we can't resolve below 9 because the hairpin opens around like that force, so you can't really see anything below that. And then we can't go anything higher than 30 just because the lifetime is too short. It's actually much shorter than our frame rate, so we can't see that. So we're really bound by those 2 parameters for expanding the dynamic range of this. And so, you can look into a bit more detail to show that, for example, the average force along the edges are a bit higher than what's on the inside. And to kind of visualize this a bit better, because it's actually really hard to see, what are the distribution of forces. You can sort of see it's more red here and then less red here, but like, it's hard to I guess quantify this or visualize this a bit better. So what we did was we just super resolution, but we downgrade the resolution to smaller grid so we can actually quantify things a bit more clearly, right? And so we get the average force of every grid, and that's a map here. We can see higher forces on the edges, and then lower forces kind of in the middle. So kind of just like as a kind of a qualitative validation of this is what we expect to see. And so of course you can do this by chopping up your movie into shorter time segment and perform the same analysis. So you get simultaneously some quantitative stuff over time. And so we from this work we can get a spatial resolution of about 30 nanometers, temporal about 60. That's kind of how long we have to look into each trace to get enough events so we can get a confident force value. Force, we can get something between 9 to 30 piconewton, and this is all in live cell, right? So this is kind of an example of using a binary force probe, but getting a non-binary kind of force dynamic information out of it. Okay, so that kind of solves part of the issue, but another thing that we're asking ourselves is, can we image tension at the time scale of collective behaviors, like hours or days, right? Because one of the biggest challenges, of course, when we use DNA is DNA's degradation, and so Irving is the one in the lab who's been pushing this. And so, we know there's a lot of different approach, like people use, PNA, LNA, modified nucleic acid to prevent degradation from happening. This is the first example of us being cheap is because these probes are too expensive to synthesize, and we can't afford them. And secondly, it's because a lot of the DNA mechanics are characterized for DNA, and once you start to modify a certain aspect, it will be useful to kind of revalidate, but the kind of knowledge about that particular DNA modification has to be built up before we can be fully confident about what are the forces, energy, lifetime, and so on. And so we're like, what can we do? We in our system we have free floating DNA probes like the ones for imagers, and we have to have immobilized ones on the surface to actually do the force reporting. And so we're like, okay, why don't we just throw in more DNA so that they serve as kind of a decoy for the target DNA that you're trying to protect, so that the nucleus is kind of occupied just eating up those things. And so it works pretty well. So we're doing here, this is some TGT experiment. So if you don't, so the decoy DNA we add is just like a 48 nucleotide double-strand DNA of whatever sequence. Um, and so it's really, really cheap to dump into the solution. And so if you do it without any decoy, TGT experiment, you can get something like this, right? So you see the background is generally high. This is because when the DNA is degraded, you're left with the fluorophore on the surface, and so it's pretty bright. And also you get like the membrane-bound nucleases that create all sorts of patterns that's not related to tension. But if you add a 5 micromolar of the decoy in the solution, and then you do your imaging, the image are cleaned up significantly, right? So the background is lower, and under the cell you can clearly resolve things that are mostly due to adhesion structures and not so much from the other membrane nucleases. And so you can clean up the molecular beacon experiment as well, right? So we did this experiment with confocal. So the molecular beacon gets chewed up pretty quickly, actually. And so after an hour, you basically see the whole background in a confocal slide just light up, right? These are just degraded molecular beacons. But then if you add decoy, like everything cleans up. So that's nice. So you can clearly see the detention signal. You can do this for single molecule imaging. There are a lot less background stuff, and when you do super resolution reconstruction, this is much cleaner compared to something like this. So the background level gets reduced quite a lot. In terms of both the localization event and you still see some bound stuff. These are just kind of the molecular beacons that's not in the bright state, that they're sometimes bound to the surface, but they can be very easily excluded from the reconstruction just by filtering by intensity. And then we tested how long this can last. So this is experiment using the DNA hairpin probes. And what you see is that without the decoy DNA, number one, all the cells are detached. And this is just because, we have RGD on DNA, and once they're degraded, there's no more adhesion molecules on the surface, so all the cells bought up and died. But if you add the decoy, then you have quite a lot of healthy cells still maintained, stuck on the surface. And this you can basically have this force probe on the surface after 24 hours and still retain images like this with the molecular beacon plus hairpin probe system. So this is quite nice, and then I think Irving has shown in the paper that he's able to just extend this beyond 24 hours to 48 hours, we just keep replenishing this DNA in the solution, right? So for that, it's a very simple method that works, and it costs less than like a dollar per experiment. I think it's like 50 cents that he calculated. So it's much cheaper than the DNA that you have to put on the surface. So it's a pretty effective generalized strategy. So you don't have to care about exactly what kind of DNAs you're trying to protect against, right? Because as long as you're trying to protect double-strand DNA, you throw double-strand DNA. If you're trying to protect single-strand DNA, throw single-strand DNA. So yeah, so that's that. Okay, so the next part of my talk, I want to talk a little bit about getting, you know, programming novel DNA nanomechanics. So we've been using a lot of DNA duplex structures and things, and so those are very useful. But can we get more novel DNA nanomechanics out of other type of structures? Is what we want to explore here. And so we have several kind of ongoing stories. We first is the we create a DNA-based catch bond. The second one is about mechanical anisotropy, and the third one is about a topological knot. I'm not going to focus too much on the latter two because those are still kind of developing stories. But I will mostly focus on the first one. And so, the first one really is a way for us to demonstrate how we can create like a new mechanism to control molecular adhesion, and maybe as a new material to reprogram the cellular interfaces. Whereas the second and third one is how can you control the stiffness of a molecule, and how do you quantitatively translate energy landscape engineering and transition pathway engineering to create the behavior precisely as you want. And the third one is kind of a more blue sky research to kind of understand some fundamental aspect about DNA conformations and stuff like the molecular friction, topologies, knots, and so on. So anyways, I'll talk mostly about the first story about our artificial catch bond that's been published last year. And so this has to get into kind of a story between slip bond versus catch bond. And I think a lot of you guys already know what the differences are. Classic example, Velcro is a slip bond, and this Chinese finger trap is a catch bond. But microscopically, if you look at this video on the left, what you see is essentially, as things dissociate, these hooks like they deform, and then after they deform to certain degrees, things slip out, right? Whereas in the catch bond, as you pull this, the structure actually squeezes onto your finger that makes it harder to break. And so really, like if you think about the general type of behavior is that force will cause some kind of molecular deformation. And if this deformation causes things to dissociate more easily, it's a slip bond. If it becomes harder to dissociate, it's a catch bond. And so I think another way to look at this is from the energy landscape or the lifetime, right? So if you have a slip bond, the higher the force you apply, the greater, sorry, the less time the bond will last. But for catch bond, if you apply a greater force, then the lifetime becomes higher, right? And so if you plot the lifetime as a function of force, slip is going down, catch goes up, right? And so there are several different kind of catch bond. There's the catch slip. So catch bond, eventually the force goes, as force goes up, lifetime goes up, but then eventually it has to enter a slip because eventually everything breaks, or a slip catch slip. And so this is actually catch bond is actually really, really important in mechanobiology. My student Micah just did a brief literature search and found like a lot, and I think Chengju also had a recent review paper that kind of talked a lot about a lot of these different catch bond. But it's a lot of those are involved in mechanics, adhesion, motility, host pathogen interactions, immune activation, mechanotransduction. And I think that the general idea really is that there is a force-induced stabilization of these bonds that creates a greater lifetime and specificity that makes it very, very useful biologically. And so, what kind of prompt us to do this is that, while DNA is really awesome for studying adhesion, the problem is that the duplex itself is too simple, right? The duplex is a slip bond, whether you're in the unzipping or shearing geometry. So how do you better mimic interactions between cell and environment? If you want to solely rely on DNA as the interfacial kind of molecule, right? So then we have to create something that's more similar to the biological stuff that has catch bond behavior, and also just to expand the nanomechanical repertoire of DNA. So the question we ask is, can we create an artificial catch bond with DNA and be kind of quantitative and rationally design this? And so it's a question that my student Mike and David took on. And so the conceptual design is something like this. So all of this still rely on kind of the mechanics of duplex DNA. But if you imagine you have a molecule like this, you tether this molecule at this point and that point, if you try to pull them apart, you see that the force is acting on this bond and that bond, and ultimately you're creating two unzipping hairpins in series, right? And so there are two outcomes. One is that the blue one would unzip, right? And so if it unzips. And so, but if the red one unzips, uh, this whole structure is still intact, right? But that unzipping would change the structure from this to that, and changes the blue part from a unzipping to shear. And so in this pathway, then your blue part would then undergo shear. So you have a pathway that's weak because it's unzipping, and a pathway that's strong because it's shear. And so if you can then create a way to select the pathway so that when the force is low, you undergo the weak pathway, and the force is high, you go to the strong pathway, then you can create a behavior that's mimicking a catch bond. And how to do that is to tweak the sequence of these two hairpins so that their force-dependent lifetime would intersect at some predetermined force here. Right? So what this means is that if the red one is we would call it the jaw, you guys will see it very quickly. It's like a fish hook jaw fishing type of analogy. And then the blue one is this one. If they intersect here, that means if you apply a force to this structure, anything below the crossover point, the hook would have a lower lifetime than the jaw, right? So if it has a lower lifetime, this one would unzip first. So that will go to the weak pathway. And vice versa, when the force is greater, the red one would unzip first, will go to this pathway. And so now you basically have a force-dependent pathway selection. And then because you have this, then you have a pathway-dependent dissociation kinetics. And so now you have essentially building different pathways for the molecule to dissociate. So here's a detailed design of the molecule, and the experiment we do is this. We did it on an optical tweezer. We call this the hook. This is the fish, and so you bring them together, and then the blue part would anneal. This is when the fish bites the hook, and you basically have a structure like this. And what happens is that the weak pathway is the unzipping of the hook, right? So the fish flies away, not flies, swims away. Or the strong pathway, when the jaw opens, well, it's the bad analogy for jaw, but anyways, its jaw opens and then it becomes a shear, and then the shear happens. And so there are very different signatures for both pathways. For the weak one, you'd expect a force extension curve to kind of just have a single low force rupture event. For this one, you have two events. One correspond to the unzipping of the red part of this not hairpin, but like unzipping of this duplex, which is shown up here, and then the hook would then shear open, and then that shows up as a very high force event. And so if you do this experiment by bring the beads back and forth very often, you see, these events, you see that there are generally two populations. One is the high force population, one is a bunch of low force population. And if you look at this in a bit more detail, you see that if you look at all the successive tethering traces, you see that it's basically fluctuating between these two opening events. One is low force, and then the high force ones would always contain a jaw opening event in there. So kind of is a validation to show that this molecule has two different pathways to dissociate. And then we showed that the dissociation is exactly as we expected. The length of the molecules all make sense. And then another really important validation is to show that this is indeed a catch bond behavior. And one of the signature for catch bond is if the loading rate changes on this catch bond, you will see a shift between the two populations, right? So if you have a force ramp, like a loading rate increase, you will see more of the high force rupture event. And if you're loading very slowly, you'll mostly see the low force rupturing event. So we would expect to see if we kind of pull this molecule apart, you will see a change in the population between the low and high force population. And that's exactly what we saw. We did some simulations based on the best knowledge of energy and stuff, and we did experiment, and you can see that at low force ramp, the low force population is higher, and then at high force ramp, the high force population is greater. And we can compare this to our theoretical curve, and we did it for three different catch bond designs to show that like they're generally following the same trend. These two align very well with our theory. This one not so much, but I think it's in the fine tuning of a lot of those energy models for DNA hybridization. But the idea really is that, with this, we can create a kind of a DNA catch bond. And ultimately, the goal is, can we use this to interface cells so that we don't have to rely on slip bond? We can now introduce catch bond for creating the right type of behaviors. Okay, so very lastly, for the next few minutes, I just want to introduce a kind of our most recent work on some kind of massively parallel single molecule assay that combines force with fluorescence. That's also really cheap and easy to implement. So it's been a group effort for the past couple of years. And so you guys all know that there are a lot of different single molecule techniques for pulling things apart. There's optical tweezer, AFM, magnetic tweezer, and there are new ones like acoustic force or centrifugal force. So the question is, why do you need another one? Well, of course, cost is one of the thing, but one of the biggest challenge, if you look at all of these techniques, is that they either use bead or they use some kind of large probe, right? And the problem is always in the calibration and reproducibility of these things. Uh, in primarily when people use bead, the biggest problem and variation comes from the bead size. You can never get exactly the right perfectly monodispersed size. You can get as close as possible, but you don't really know exactly how big or how small they are. And so when you do this, very often people have to still do individual calibrations of every bead, and that's a very long process. And a lot of times, those processes result in low throughput or a kind of failure of your molecules. And so that's one of the problem. The other is multi-tether, right? So, if you have one tether versus multi-tether, like you have to find a way to piece them apart, and that ultimately depending on the surface density and ultimately the throughput of your technique. If you are not careful about surface chemistry, which even when you're careful, things don't work. Your bead will just stick to your surface depending on what kind of system you try to study. And getting the surface chemistry right is very, very tricky, and it's more magic than a few than science. And even in the ideal scenario, you have a single tether with a target in there. You're bound to have the tethers effect kind of convoluted with the target molecule you try to study. So you gotta all your energy stuff gets kind of blurred out, right? And so you can't get exactly the behavior that you want to see. And so this is all because the bead is one that's both doing the force application and also position detection. And so that also limits the response speed of these techniques. And so instead, what we do is, let's just get rid of the bead. Let's just use a very large piece of single piece of double-stranded DNA as our force applicator. So if you put this under a shear flow, what we basically show is that if you have a large enough piece of DNA, that DNA can generate enough force to actually unfold molecules. And by doing so, you basically allow you can then just put this on your microscope and use a single molecule fluorescence method as your detection method for conformation changes because you can do localization of the molecule. And so the advantage of this is that your DNA molecule, you can PCR this, and so the length is precisely controlled, right? So you no longer have that bead size variation. And it's intrinsically single tether, right? Because you have one molecule attaching to a bunch of stuff to the surface, and you never would have problems with multi-tethers. And it gets rid of a ton of problems that's associated with the bead. And in terms of detection, we can get 2 to 5 nanometer resolution if you do localization, or a single molecule FRET to get sub-nanometer resolution of the distance. And the another really cool advantage is that your molecule is not on tethers. Your detection is not on convoluted with the tether. You're detecting the true position of the molecule. Um, so here's just a kind of some experiments showing, kind of stretching DNA under flow. You can label the whole DNA with flow force, intercalators, and you can see them like this, little, it's kind of like DNA curtain, but like randomly on the surface. And you can have the terminal flow force. You can basically see where they are being stretched, and there are pairs of those. And so some of the experiments you can do is to look at irreversible ruptures. So you can basically build a chymograph of these molecules, and you can detect the rupture event very clearly, build a histogram, I guess, event survival curve, and force histogram from this, all like within one experiment. And then you can also, for example, do label this DNA with terminal flow force, but also, a DNA hairpin. If you label at one end, you can look at how this end moves as you kind of apply force to this molecule. And so this is led by Orion, and then you can see if you increase and decrease the flow rate over time, this terminal flow force gets stretched. And if you look at one frame, you can't really see much, but if you do a single molecule localization, on this end, if you do a force ramp, you see the position of this goes further and back, right? As you expect, as you kind of stretch this piece of DNA with greater force. And then if you look at this one, at this end, you see two populations. One correspond to the folded state of the hairpin, the other the unfolded state of the hairpin. And if you could look at this over time, you see, this is the unfolding event, and then you see the refolding event. And then from this, with our force calibration, you can then build a force extension curve like this, right? So you can unfold and then refold, and then pay attention to this. Like this is vertical, right? So normally when you think of a force extension curve, you have a tether. So all of your force extension curve is kind of a nonlinear elastic chain, right? And you see a rupture event. But here, this is really the true end position of the molecule. So it doesn't move and then it unfold and then refold, right? So if you want to study something that's decoupled from the tethering effect, then this will become a very, very powerful tool to do that. We validated this with optical tweezer measurement to show that the forces are as expected. And we also did this with FRET to show that if you have a hairpin and the transition distance is too short to be resolved, you can still see it by single molecule FRET, right? So you can see the sudden change in the rupture, the unfolding force, and the refolding event. Yeah. So anyway, we hope with this we can have a better throughput to screen a lot of molecules, especially if we're trying to create novel DNA nanomechanics. Our models are not very good in general in predicting nanomechanics. We're okay, but not like perfectly good, right? So we need more data, empirical data, so we can find a model that's better suited, and also just for validation purpose, because very often you will see in publication that they want you to validate what is the fluorescence force response of your molecular force probe. They want empirical, right? So this becomes a very easy way to directly get that information out, because you have the force and fluorescence at the same time. And so, yeah, so with that, I just want to thank you all for your attention, and my group and funding agencies for supporting this work. And I'm happy to answer any questions.
Amazing. Let's see. I know Khaled put some questions in the chat. Maybe we start there if nobody
Uh,
else has a question.
right. Okay. Yeah, I can read the questions here. Just give me 1 second. So Khalil said, how do you distinguish between force-mediated dissociation and termination of tension or dissociation of ligand receptor? Is this
It
regarding
had
the
to do
first
with
part?
the kinetics of the molecular beacon paint.
Right. Yeah. Uh, the short answer is we can't. And so, you get a time series of binding event at this particular location, right? And so, like you will see event, 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 and you see like nothing. And so you don't know if that nothing is because there's still tension on this molecule and it's just nothing painting, or is because of the receptor is completely dissociated. So we don't have a good idea about that. But statistically, we can see that, like for example, like there is a cluster of events here, and then there's nothing on a time scale that is beyond the normal kind of k on. Then you can sort of separate those two as separate events. But there is no way to decisively say, this is the point when the tension has disappeared. There's always some ambiguity to when the tension disappears and when it reappears. And also that relates to the whole temporal aspect of the kinetics, right? If your tension varies very, very quickly beyond the rate of the detection, then you're really just gonna get an average. You can't get instantaneous tension here. Oh, and then the next question is, what is the Km of DNAs and target? Um, to be honest, I don't have a good answer for you. Right? We kind of just showed we didn't really care about the DNA's part of it. We just say if we do this, can we see things better? Uh, do you have some insight on this?
Well, no, I mean, I was just asking because I think in your slide you said you use 5 micromolar, and it made me think you're probably saturating all the nucleases, and so you have a concentration that exceeds that Km,
Yeah,
right?
yeah.
But I was wondering where that Km is and whether it hovers around that 5 micromolar, and that's why you chose that concentration.
Oh, it was just whatever is convenient. 5 works well. If you go lower, then I think it doesn't as well. So in our paper, we have a bit more data on like what are the protective effect for different concentration. And so maybe some information about KM can be extracted there. But yeah.
Yeah, since I already have you, I'm gonna take this opportunity and ask. I mean, this is all really cool. Do you have you integrated your catch bond system with cells? And if so, do you think you would see an impact, just like unzipping versus shearing mode TGT drives
Mm.
different biology? Could a catch bond forming tether drive some biological responses?
Yeah, that's a great question. Just before we published it, there's another group who also did a catch bomb with DNA with a different design. They didn't do the single molecule characterization, but they did the rolling adhesion. So the rolling adhesion is kind of a very quick test of because it requires catch bond for a stable rolling to happen. So, you can do that, but it's been done. But if you think about for other type of cells, right? I think it's really about if you have, for example, a certain receptor ligand interaction that are not catch, if you turn that into a catch, what would happen? Right? So that's kind of an interesting question. And then the other one is, when we put our, for example, TGT with like a catch bond on the cell part, like that interaction is still catch, but then the TGT kind of modulate that catch bond and make it a bit like weaker or something, right? But then so the modification has a certain limitation to that. So really, I guess one of the question is, can we instead of modulating and making it weaker, can we replace, right? Can we replace a catch bond, like, either it's integrant or cadherent, replace that, like stop the function, replace it, but with catch, right? So there was a study a while ago looking at E-cadherents, and then they basically put a snap tag at the end of E-cadherent. So that knocked out the E-cad actual interaction because it's not interacting. And then they replaced that with DNA. But they did that with just like sheer DNA, right? And so the ECAD interaction there would be kind of an entirely slip bond. So if you turn that into catch, would you see differences? And I think a lot of this are subtle differences, but I think subtle but important. I don't know the answer. We haven't done this to biological systems, but if you guys have a good system, we're more than happy to supply the designs.
I have a question about kind of going back to the single molecule, the different forces that you're able to detect. Did you identify any molecules where you actually saw like a change in the force over the lifetime of the experiment, where the molecule maybe had like 9 piconewtons of force and then it increased or decreased?
Are you referring to the last part or?
No, like the first part where you had the
Ah,
paint imaging and you're able to actually like define the force on the molecule by the binding kinetics. Did you see
right,
any
right.
single molecules where the binding kinetics like changed in the middle of the imaging, or is that too much data to try to get out of it?
Yeah, that's a really good question. Like when we started the project, we were like, oh, it'd be great if we can see like a transition of this molecule, like from like slow kinetics to fast kinetic. Then you can see, oh, the force is increasing, right? And so unfortunately, it's really hard to like because it just depends on how you process that data. Let's say you have one molecule and you see events. You can bend it in ways you want and then say, oh, what's the average force and how did it change? And so your bending kind of changes how you interpret, right? So we didn't go too much detail because we have no confidence in how to bend that data. So you can sort of, but and then we also didn't have an exact biological question that we want to ask with that. But you can imagine if you have a trajectory, you can say, okay, what's before and after, like first half versus second half. Then I think that would be a meaningful question. For us, it's really about, for example, fine-tuning how many events you get. Like if you only get 2 events, you have no confidence in the average. You have to get like 5 to 10 and be like, okay, within this, right? But then if they start to change, you're like, oh my God, is this because forces change? Or is it just like statistics, right? And so that part is a hard part, but I'm hoping that maybe this is a kind of a way for people to think, maybe there's kinetic information, and then can we do better kinetic encoding so that we can actually get better information out of this system? So great question. I don't have a better answer than this, but that's the limitation.
Yeah, I think it's really fascinating, thinking about maybe even an individual focal adhesion and just well, first just spatially within that focal adhesion, is there a distribution of forces? But then like maybe a focal adhesion that's forming over time, do you see changes as like this integrin, maybe it's like one of the first ones in the nascent adhesion and it's still on in the mature adhesion, and did its force distribution change as
Yeah, yeah,
the
exactly.
proteins inside the cell are changing? I think,
Yeah.
yeah, there's lots of pretty exciting things you could try to
Yeah.
try to look at.
Yeah, and please, if you guys have interesting system to look at, we're more than happy to share our code and stuff too, so you can look into those processes.
Awesome, thanks.
Yeah.
I think I see a hand up, Elizabeth.
Yeah, hi. Very cool stuff. For the probe that, I guess the flow probe, the significance of the tether makes sense because you kind of need the counterforce, but would it work, I guess, on like a cell membrane too? Or would the
Mhm.
cell have to be stationary?
Yeah, that's a great question. So, I guess what you're imagining is if you put those probes, like it's like having hairs on the cell, and you kind of
Yeah.
float. I think it theoretically they will work. The shear that we put in there is very high. So what I worry is that you actually shear your cell off. And then there are also like, cells are mechanical sensitive to shear stresses. They're like membrane receptors to those, right? But if you can, I guess, I guess isolate these effects or look at cells that are normally in high shear environment, maybe that's okay, right? Like if you look at epithelial cells or endothelial cells in the blood vessel, like maybe that's okay. And so I think it is possible, but just double check that the shear is not too high.
No, yeah,
You
that's
can also
exactly
change that by kind of making a longer DNA. Like here we use 16 kilobase pair DNA. But if you increase the length, you don't have to increase shear to add more force. So that's also possible.
very cool. Thank you.
So as I have a quick question about the catch bound, DNA catch bound. Yeah, as you mentioned that there was a paper before your paper, unfortunately, in Nature Chemistry, a little bit earlier than you guys, probably generated a little bit trouble for your publication, hopefully.
Yeah.
So I wonder, I know the design is different. I wonder what's your opinion on the design difference in terms of the molecular behavior difference? What's the implication? I can say there's some difference. In fact, personally, I would think your design almost have certain advantage over their design for particular applications, at least, because their
Mhm.
design. I mean, anyway, I'll hear your opinion first, I guess.
Yeah, of course. Uh, I think both designs are basically looking at how to create two pathways, right? And then the two pathways have different dissociation kinetics. I think what ours is that we have much better model for unzipping, so you can design this very quantitatively. So we explored a lot of parameter space for this, and then we can show that they work. I think one of the primary reason is that shear is not as well characterized as unzipping. So where like how do you predict the behavior may be a little bit harder. Um, uh, I think their design, there's nothing wrong with their design. It's a little bit more complicated in the sense that there are a little bit of things like you have to have a gap between the shear construct, and so how does that, like there's a lot of unknown about different DNA structures that have defect in them. What are the nanomechanics? So I think qualitatively it works. Quantitatively, can you predict and design? Maybe not as rational. One of the advantage they have is their construct is shorter. Ours is big. And so we're now working on Gen 2 and Gen 3 of this catch bond. So now we can get things a bit shorter. So hopefully that bridges the gap. Yeah.
So what would be the challenge you're facing if you want to make it smaller?
Right. So right now, like you see that massive loop, right?
Yeah,
That
yeah.
thing is very hard to do, especially like for people who want to incorporate this stuff in a hydrogel. Like it's just too much synthesis for the scale they want. And so
So is it necessary that the loop has to be that long?
yeah, this one's necessary because the red part is very long, and so to completely unzip this, you need a large enough distance for this particular design.
The red
And
part
so
need to be long because you want it the binding to be stable. Is that why that has to be long?
this is because let me see if I can find a picture. Right. So you see this, that's the loop. Like
Uh-huh.
this loop is here. So in order for this DNA to completely unzip, like that end-to-end distance has to be large enough.
Mhm.
And so this loop is almost the size as like this plus that in order for a complete unzipping to happen.
Yeah. Okay, I don't want to take too much of your time. Really interesting design. I like the catch bound study. And I think one advantage from my perspective might be your design, in their design, right? So I think that it requires some formation of new DNA hybridization. So that
Yeah.
could cause a delay in response. I don't know
That's right.
whether
That's
that
right.
will be what implication that will be, but at least from design point of view, yours is kind of instant. Is there? There's it requires formation of new DNA base pairs.
Right.
There could be
Yeah.
delay. Okay.
Yeah.
Yeah.
Yeah. Yeah.
Great work.
I agree with you. That's right. Yeah. Good point.
All right, awesome. I think we're a little bit after 12, so I think thanks, Isaac, for an awesome talk. And, I think you presented so many exciting new tools, and you didn't disappoint me on the cheap and doable aspect either. So, but some of those are, really the most exciting. I definitely am somebody who's suffered from having my cells eat my DNA tension probes.
Yeah.
And the idea that I could just chunk in some DNA and solve
Yeah.
it is amazing.
Yeah.
So-
Double check the immuno stuff, like, if DNA causes anything. But in our case, I don't think it depends on the cells you use, but double check. Yeah.
Yeah, no, but it kind of goes from like, oh, we can make those PNA probes to something that's actually much more doable. So
Right. Yeah.
really,
That's
really
right.
cool.
Yeah. Thank you.
All right. So thanks so much for being here. And I guess we'll see everybody again next month, right, Alicia?
Yep, next month.
Okay, great.
All right.
Bye,
Yeah,
everyone.
Take
Bye.
care.